Hi there. In this video, I'll be talking about gene duplication events, including whole genome duplications, which each have led to rapid evolution in many cases, and have led to the formation of multigene families, which have led to quite a few evolutionary innovations. Multigene families are sets of genes that originated from an ancestor gene by duplication and divergence, so that each of the derived versions have evolved different functions, sometimes very different functions. The new functions can allow rapid evolution of species. Some new gene versions have only slightly different functions, while others can have vastly different functions. And so many evolutionary innovations and changes have been the result of these duplications. So these changes can be fairly rapid because instead of starting out to evolve essentially a brand new gene, the genes can be duplicated. One gene can remain the same, and the other one is free to mutate and change. And so you can get rapid changes from these duplication and divergence events. Here is a short list of some multi-copy genes, including actins. Myosins would be in this category also, in muscle cells and in cells in general. Eggshell proteins, many different versions of the eggshell proteins, alpha and beta globins to carry oxygen throughout the body. Heat shock proteins, histones, over 75 different versions of those. Immunoglobulins, the variable region, over 381 versions of those. And then we talked about the ribosomal RNA genes, the tRNA genes, which are in multiple copies also. And there are literally thousands of multi-copy gene families. All multi-copy genes in gene families started out from a single gene, such as this one. Then at some point in time, there was a duplication of that gene, either from recombination or a loop out of one piece of the DNA followed by DNA polymerization or some other mechanism. And so one gene retained its function, and the other gene was free to mutate and take on slightly new functions. So eventually, the gene that was free to mutate took on functions that set it apart from the original gene, so that it was now what's called a paralog, that is, a gene that arose from another gene but significantly changed. There can be subsequent duplication events, including some that create new versions of the gene and some that create pseudogenes, which are portions of the original gene that no longer function. Some are not ever transcribed, while other pseudogenes are transcribed but form a non-functional protein. Also, some of the genes can be moved to other chromosomes, again by recombination events usually, or transposition events. And again, some of these genes can have functions similar to the original gene A, while others can evolve quite different functions, but you can always tell that they originated from gene A. One interesting multigene family is the one that includes a large number of genes to create flagella in bacteria. So flagella can be of many forms, either coming out of one part of the cell, coming out of multiple parts of the cell, being just on either end, and some coming out everywhere on the cell. And they're primarily for motility, to find food, to move towards food, escape anything that wants to eat you, a predator, or to aid in moving food particles and other things towards the cell. In gram-negative bacteria, such as E. coli, there are two membranes, and the initial portion of the flagella go through both portions of those membranes. And then there's a motor mechanism down within the inner membrane of the cell. And that twists the long filament of the flagellum, propelling the cell through the liquid that it's in. For a time, people were having trouble reconciling how something this complex evolved in a bacterium, until a study was done looking at the proteins and genes involved in producing a flagellum. And it was found that it proceeded in many steps, and furthermore, they could see that the evolution of each one of the versions of those genes, as well as the construction of the flagellum, occurred from the inside of the cell to the outside, to the tip of that flagellum. So as it's constructed, it starts on the inside of the cell. It builds outwards further and further, and that also follows the evolution of each one of those genes as well. So evolution and construction of the flagellum, which originally wasn't a flagellum, it was only later that it became a flagellum, and I'll show you that in a few minutes, began with this ring structure here, which could spin. And next, these moat molecules evolved from that same system, and those caused the movement of a circular movement of that ring structure. There were other proteins and genes that produced the outer ring, and finally later genes added the hook, and then other genes followed, producing the filament and eventually the cap on the end. So that all occurred evolutionarily in that direction, and developmentally also in the same direction. Furthermore, the genes are located in clusters, and the most related genes are in the same cluster, but then in different places on the chromosome you have different clusters. Each of those clusters then contributing to building of those flagella. And if you look at these gene clusters, you can actually compare the sequences, both the DNA sequences and the protein sequences, and determine the evolutionary pathway that was taken to create these genes and then build up a flagellum. There's a very nice animated movie on YouTube, here's the address down here, which shows how the flagellum in an E. coli is built up. And here is these are just pictures out of that same video. So first the inner ring is produced, and then another cone is built around that. Eventually those hook up with the motor, and then an outer ring to go through the outer membrane, finally adding to what's called the hook of the flagellum, and then eventually the filament is added after that, becoming longer and longer and longer, depending on the species. And so here's a picture over here showing that. Here's the inner ring and the moat proteins, the motor complex, and then the outer ring, the L ring here, and then eventually the hook, the junction to the filament, and then the filament itself and the tip at the end. And again, the first genes to evolve are here, and the last genes that evolved are way out here, and developmentally it follows the same pathway. Now, the flagellum, as it stands today, was not always a flagellum, and it is clear that it evolved slowly as part of a different system. And so here's a passive pore, which added to part of that, and then a gated pore, and then an ATP synthase, and then an export system. So this was a secretion system to secrete certain proteins and molecules out of the cell originally. And then the outer part became more and more elaborated, eventually with a pilus and with a cap pilus, cap, and rod, and then the ATPase was replaced by the moat proteins. And ultimately, this filament got longer and longer to become the flagellum. Archaea also have flagella, and these have also evolved in a similar fashion as did the bacterial flagella. Here are different organisms. M means modal, meaning they have flagella, and then the non-modal ones are down here, which had flagella but evolutionarily lost them because they were unnecessary to those particular organisms. And as with bacterial flagella, the archaeal flagella are in, the genes are in clusters. And if you look at the sequences in each one of those genes, you can relate one gene to another, indicating that these were gene duplications followed by divergence and specialization by each one of those genes and the resultant proteins. So you can see by this that even bacteria and archaea have very sophisticated systems, and multigene families are one of the reasons why they could evolve these functions so rapidly. Another well-studied multigene family is the globin gene family. So on your chromosome 16, you have several versions of alpha globin, and on chromosome 11, you have several versions of beta globins. And together, these globins produce proteins that carry oxygen in the blood to oxygenate the cells around the body. And the earliest version of globin is myoglobin, and it's in invertebrates as well as vertebrates. But then you have the alpha and beta globins beginning with the vertebrates, including fish, and then all the way up through mammals. In humans, as well as all placental mammals, there are several versions of the globin genes, and they turn on and off during certain parts of the developmental pathway. So beginning at conception, the first ones, the zeta, which is an alpha type, and epsilon, which is a beta type, are turned on at the highest levels. Over the next few months, those levels drop, and there's an increase in the true alpha globin, as well as gamma globin, which is a beta type globin. And by 6 months in, 3 months before birth, then the levels of the gamma start to decrease and the levels of adult beta globin begin to increase. And by birth, you have true alpha globin and then a mixture between gamma and beta globins. And then by 6 months after birth, then you have true adult alpha and true beta globin, as well as a little delta globin in the bloodstream. The reason for the different globins is that the oxygen has to be transferred from one type of globin to the other in order that the embryo and fetus are able to get enough oxygen to grow and develop. So the mother's lungs have alpha and beta adult type, and so the lungs pick up oxygen. Oxygen goes through the mother's blood and nourishes the mother's tissue, right, with oxygen. But some of that oxygen has to transfer into the fetus, and that means that the affinity for oxygen for the fetus blood has to be higher than the affinity for the mother's blood. So there's a net transfer of oxygen from the mother's blood into the fetus's blood and then into the fetus's tissue. So this is a graph showing the differences in those affinities. So here's adult hemoglobin, there's the fetal hemoglobin in red, and then myoglobin in the tissues and cells of both the adult and the fetus. So you can see that at a level of about 30% pressure of oxygen, that's essentially the concentration of oxygen in the blood, about 60% of the adult hemoglobin is saturated with oxygen, and about 80% of the fetal hemoglobin is saturated in the fetal hemoglobin. So there's a net transfer then of oxygen from the adult, which gives up oxygen a little easier, and then that delivers it to the fetus's blood, which picks up oxygen more readily. And then subsequently, for both the adult tissue and the fetal tissue, the myoglobin will pick up the oxygen more readily than either of the hemoglobins, including the adult and the fetal hemoglobin. So there's a net transfer of that oxygen to the cells of the adult and the fetus, although there's also a transfer of oxygen from the adult hemoglobin to the fetal hemoglobin in the placenta. So if these genes are examined, the sequences of these genes are examined back into the past, you find here are placental mammals where there is a separation between the types of alpha globins and the types of beta globins. There's a split back here in the marsupials, so they don't have as many different types because their fetuses or their early developmental individuals develop outside of the mother's womb and only are there for a short time early in development. You can also follow those lines back further when there was a separation on the chromosomes, and then back further during the duplication event, actually many duplication events by the time there are fish, and then back into the invertebrates for the progenote gene, some of which became myoglobins. So we can trace this forward, starting with invertebrates, which have only one copy, which is myoglobin-like. And this was present in organisms by about 600 million years ago. Next, by about 500 million years ago, starting with the jawless fish, there were duplication events. And so there were then many alpha copies and many beta copies, all looking pretty much the same. They were not that different one from another, but only one version of each is shown here. Then by about 400 to 350 million years ago, these were separated on different chromosomes. And that began about the time that some fish became lobe-finned fish, which eventually became reptiles. And one of the major differences then was fish and amphibians lay their eggs in the water. So there's a transfer of oxygen there, and they develop in that water. Whereas with reptiles, they start laying their eggs on land. So the oxygen transfer into those globin molecules is going to be different than if they were in water. So now you need different globin genes and different globin proteins in order to assure that that animal receives oxygen into its blood, and then that's transferred into its tissues in order to develop properly. By about 200 million years ago, mammals first appear, and most of them were marsupial mammals, where now there needed to be a transfer from the air to the lungs of the mother, into her blood, and then into the fetus's blood, and then into its tissues. By about 110 to 120 million years ago, the first placental mammals appear, and the demands for oxygen are greater and over a longer period of time, so that there's a embryonic set of globin genes which change over to a fetal set of globin genes which can accept more oxygen, and then finally the adult globin genes for once the child or the baby is born. So in each stage of this evolutionary pathway, there is also a parallel developmental pathway that was possible due to the duplication and diversification of each one of those genes, which led to a different protein that was capable of accepting oxygen at different oxygen pressures. Also note in this diagram at the top that there are several genes with a psi, a Greek letter psi alpha, psi zeta, and psi beta, and there are actually even more than those, and those are pseudogenes. So in each one of these duplication events and diversification, almost always you have some pseudogenes that are produced, and there are many types of pseudogenes. There are several types of pseudogenes. Here's a functional gene at the top. One type of pseudogene has an altered promoter region, so the gene is always turned off. It's never transcribed, even though the entire sequence is there. The second one is where there's a nonsense codon due to a mutation. And so you have transcription of the RNA, but during translation, there's an early stoppage of that protein, so the protein does not function. There are also ones where there's a truncated gene, so only part of the gene is actually there, and these may or may not be transcribed. And then finally, there's this one down here that looks like a processed gene already that's been reinserted into the genome. So it is without its introns, and it has a poly-A tail, so it looks like it was an mRNA that was reverse transcribed into a DNA and then inserted back into the genome, and usually these are non-functional also. So in short, some pseudogenes are never transcribed, so they're not expressed that way. Some are expressed as RNA, but then produce a non-functional protein. And a third type is transcribed, but never translated. So essentially three different types. Another multigene family is the histone gene family. Archaea do have histones. Bacteria don't have histones, but they also have DNA binding proteins. Histones are the ones that wrap up the DNA for either being expressed or not expressed, or during mitosis, etc. So the archaeal type is here. That split sometime in the past into a type of histone that became the H1H2 family and the H3H4 family. Millions of years later, H3 and H4 separated, diverged. So then you have an H4 family and an H3 family. These little cones or triangles indicate that there are many different versions of each one of those. The H4 family is the most highly conserved of the histone families. H3 is slightly less conserved, so there's more variation in that one. And then you have this split between the H2 types and the H1 types, the H2A, H2B, and H1s. Eventually, this is the type that we have and most mammals have, where they have an octamer consisting of two H2Bs, two H2As, two H3s, and H4s that make the main histone that DNA is wrapped around. And then the H1 further condenses the DNA by bringing those histone octamers together. And as I stated earlier in the table, there are over 75 different versions, different gene versions of histones in that multigene family. The different versions act differently in different organisms and also in different tissues. So you can get differential expression of genes depending on which versions of those histones are used in those cells. Although some duplication events have been caused by recombination and transposition, many of the events have been caused by whole genome duplication events or polyploidization. And these dots on this phylogram going from plants over here, where each one of the dots represents a genome duplication event. So there are quite a few in plants. Plants have used this to expand their genomes and to speciate many, many times in the past. But these events have also occurred in fungi and in animals, including the vertebrates. So over here, separating invertebrates from these are the vertebrates up here, you have a duplication event here early in vertebrate evolution. And that one line leads to lamprey and hagfish, which are called the jawless fish. They have no jaws, no true jaws. And then there's a second duplication, so now you have 4 sets of chromosomes, and that leads to gar, sturgeon, sharks, as well as amphibians, birds, and mammals. And then there's yet another duplication, which leads to the ray-finned fish. And so there's now 8 sets of chromosomes to work with, and each one of those then contains all of the genes that were on the original chromosomes as well. One set of genes that have been investigated a great deal are these Hox genes, or homeotic genes that control some major developmental processes. So if you have that ancestral chordate that's separated from invertebrates, there was one set of these Hox genes, and there are 14 different versions of those originally. But then there was a duplication event, so now you have 2 sets of those genes, as well as 2 sets of all the genes, and that leads to the jawless fish. So again, that's this event right here. But notice that after the duplication event, there was divergence, so that in one of the loci, this version was lost, and in the other locus, this version, that version, and that version of those genes were lost. And those really had a lot of effect on the body plan of those organisms. So in terms of development, that changed the form and function of some parts of those organisms. And then after that, there was another duplication of the genome, and then divergence from that. So there are now different versions, 4 different versions of those genetic loci carrying those Hox genes, and each one being fairly unique, one from another. And that's indicated by this duplication event here, and one line of that then became the sturgeon, gar, and sharks, and then the other part of that clade wound up over here with mammals, birds, amphibians, and lobe-finned fish. But then there was a further duplication, so now there are 8 copies of each of those genes and gene types in the what are called the ray-finned fish. And these are the things that you typically think of as fish: zebrafish, cod, mackerel, etc. So here is this last duplication to result in the bony or ray-finned fish. And so now we have 8 copies of those gene loci, containing, now none of them contains 14 of the versions anymore. They all contain fewer than that. And this controls the body plans of those organisms and really results in quite a varied set of body plans, going from a pufferfish to a salmon. So here's the phylogram overlaid with the type of appendage each one of those organisms has, as well as the Hox genes. So down here we have the ancestral Hox locus with the 14 versions. Then there's a duplication and some diversification leading to the ancestral jawless fish, hagfish and lamprey. And then there's another duplication leading to the jawed fish, which includes the sharks, and there's the type of fin they have. And then further up, you go to the lobe-finned fish up here, and then eventually mammals and birds up here, so that you have different patterns of each one of those Hox genes that leads to the different body plans. That is, do you have a fin? Do you have a lobe fin? Do you have a rudimentary foot? Do you have a hand? Do you have a wing? And that's what these things control. And then back here along the line to the ray fin fish, there's a further duplication leading to diversification, including zebrafish, paddlefish, boxfish, salmon, etc. So these duplication followed by diversification events, both individual genes and whole genome duplications, have led to sometimes rapid evolution and really major changes in the morphology of fungi, of plants, of animals, and many other organisms. So gene duplication events, as well as multigene families, have had a major effect on evolution and the evolution of certain lines of organisms, especially in plants and animals, but also in other organisms as well. So that's the end of this video. Thanks very much, and I will talk to you later.